So welcome to the first mini lecture that I've prepared for you for the first day of class. What I've given you here, I've called this number 1, Overview in Homeostasis, since homeostasis is mostly what we're going to be talking about. I've given you some learning objectives here. And these are things that you should be able to do or be able to understand by the time we're done. And if you want to follow along in the textbook in the 14th edition of Fox, this is going to be pages 4 to 10. The other little mini lectures, I've called them 2A and 2B and 3. So they're all very short, and I hope that you'll take a few minutes to look at them. So let's go ahead and get started then. So we want to start with kind of a brief discussion or a brief reminder of the idea that in physiology we have a hierarchy of organization, and we can go from the simplest, which is going to be the chemical level, all the way to the organismal level. At the simplest level, we do have chemistry, so we're going to have atoms that are going to be combined into molecules. That would be our second most complicated level. Molecules then can be combined to form cells, and then groupings of cells that have a similar function are going to combine to form tissues. We have different tissues that go together to form an organ. In this example, we're looking at the stomach, and the stomach is going to be composed of epithelial tissue here that lines the lumen of the stomach. We have smooth muscle here in the wall of the organ. We have a serous membrane, which is going to be a combination of connective tissue and epithelial tissue as well out here. All of these different tissues together form the organ. We put different organs together, and in this case the stomach would be part of the digestive system, working together with the small intestine, the large intestine, the liver, the gallbladder, the esophagus, etc., etc. We put multiple systems together to make a more complex organism. So within an organism, we'll be discussing not only the digestive system, but also the nervous system, the musculoskeletal system, the endocrine system, the cardiovascular system, the respiratory system, etc., etc. So all of these different organ systems are going to be important for the overall functioning of the organism. In complex organisms, we see that there are a number of necessary life functions. Fox does not discuss these, but this is kind of a list that I've accumulated over the course of several years. The ones in red are actually ones that come from different authors, and then the ones in black are ones that I kind of have included myself. So let's start at the bottom. We'll start with growth and differentiation. All living organisms have to grow. We have to have differentiation of cells so that you can have different functions, as we just saw on the previous slide. Living organisms have to be able to reproduce themselves, and not just sex, which everybody wants to talk about, but you think about reproduction of cells by mitosis to replace damaged cells. We can kind of lump metabolism, digestion, and excretion all together. Living organisms have to have a way to bring in nutrients. They have to have a way to generate energy. They have to have a way to break down complicated foodstuffs or complicated molecules into simple molecules that they can use or store to produce energy. And then they have to have a way to take the trash out. So they have to have a way to eliminate products that are left over or products that don't have any value to them. Additionally, living organisms have to have a way to move. And I have here movement. I don't mean just movement like locomotion within an environment, but we also mean movement of substances within an organism. So when we talk about the cardiovascular system, we are going to be talking about the movement of blood through the blood vessels. When we talk about the digestive system, we're going to be talking about the movement of foodstuffs throughout the different components of the alimentary canal. Additionally, living organisms have to have a way to respond to stimuli in the environment. Now, if you remember any discussion that you may have had in the past about the autonomic nervous system and the fight or flight responses, this makes perfect sense. So if you happen on something that is frightening or scary, you can run away from that threat, or you can stand up to it to respond to that stimulus. So again, we mean not only stimuli that may be in the environment around us, but also stimuli in our internal environment. We have to have a way to produce changes in response to that. And then additionally, maintaining boundaries. And I say maintaining boundaries not only at the organismal level, where you could see that in the function of the skin, but also even at the cellular level, where the plasma membrane serves as a fabulous barrier between the cell and the extracellular environment and regulates pretty tightly what goes in and out of the cell. Okay. Now, in physiology, the main principle that we are going to be coming back to over and over again is homeostasis. Homeostasis. And when we talk about homeostasis, we really mean this balance in the body's internal environment. Fox uses a term, he calls it internal constancy, which I really like. And it's the idea that there are, for different variables, certain set points or certain constant levels of these different variables that the body works to maintain. I've given you 3 examples here: body temperature, blood glucose, and internal pH. Moving too far away from a set point of any of these values then can produce damage, and which can ultimately lead to dysfunction. You can produce damage or dysfunction at a cellular level, at a molecular level, and that can be relayed to the organism, to the organ level, to the organ system level, and then to the organismal level as well. Now I've got here 2 different little links here. This bottom one, this YouTube link, is a newsreel from the 1930s, I believe, and it is actually Helen Keller. And she is speaking with the help of her teacher, Annie Sullivan. And you may remember that Helen Keller lost her sight and her hearing when she was a baby because she had a high fever for a very extended period of time. This second link is a little bit more, it's a blog, it's not a newsreel here, Cooking in a Sauna, but this particular author outlines an experiment done by Charles Blagden in 1775 when he had a room that was heated to a temperature that would be sufficient enough to boil water. He had several helpers in this room with him. He had a dog, he had a steak, and he had some eggs. And at the end of the time in this very hot room, what he saw was that the steak and the eggs got cooked, but the helpers and the dog had an elevation in temperature, in their body temperature, but they did not cook. Why did they not cook? They didn't cook because they could maintain homeostasis. All right. Now, in maintaining homeostasis, then, we have to talk about the body has to have a way to monitor these conditions and then produce possible changes here. So these different variables are going to be monitored by the body, and the component that actually detects changes here would be a receptor. Receptors then can detect changes, or they can detect things like chemicals or hormones. They can be receptors for stretch in a vessel wall. They can be receptors for body temperature. Really, receptors can monitor practically anything. The control center here is going to be, or an integrating center, I think is the word that, the phrase that Fox uses. This is going to be the portion of the body that receives signals from the receptors and determines whether a response is needed. So it monitors the internal environment and determines if we've gotten too far away from that set point that's been established. The effector is going to be something that brings about a response. Fox describes the effectors as being the defenders against changes in this internal environment. And you should be able to see that we have kind of a flow of information, correct? So the receptors detect a change, they send a signal to the control center decides, quote unquote, if something needs to be done and sends a signal to the effector. All right, let's use a non-physiological example that you may be familiar with. So the temperature in your apartment is increasing. You've got your thermostat set to 78 degrees Fahrenheit. And when the temperature rises above 78 degrees, that signals the AC unit to turn on. The AC unit begins to cool, and the temperature in your apartment then begins to fall. Okay, so if we were to plug these events into, this is our flow of information that we just saw in the previous slide, with our receptor sending signals to the control center sending signals to the effector is bringing about a response. Here, this is our flow of information. Then we could assign different roles to these different events, or these different players that we saw previously. So the rising temperature would be the stimulus. We could identify the thermostat as being the receptor, and we also are going to identify the thermostat here as being the control center, because the thermostat is determining if the temperature is too high or not. The AC unit would be the effector. and the response would be the change or the decrease in the temperature as the temperature begins to fall. Okay. How long does the AC unit stay on? The AC unit stays on until the temperature falls back to the set point, which we identified as being 78 degrees Fahrenheit. Now remember too that the change that is brought about by the actions of the effector here is actually going to be detected as well by the receptor. So we can think of this change is also going to be detected. The change feeds back into our flow of information. And we would call this negative feedback because in this particular case, the response is negating the initial stimulus. Our initial stimulus was an increase in temperature and our response is a decrease in temperature. Let's look now at a more physiological example. This would be the regulation of blood pressure. This particular author is identifying then, he's sort of plugged in our receptor, our control center, and our effectors here. And in blood pressure, we've had some change in the condition where our blood pressure increases. The receptors that detect this increase in blood pressure are actually baroreceptors. They're detecting stretch in the blood vessels. These stretch receptors send signals to the cardiovascular center that are located in the medulla of the brain. And notice here that the medulla of the brain are sending signals to multiple effectors, not just back to the blood vessels, as you might expect, since that's where our initial receptor was, but also to the heart. And the net result is going to be that the heart beats less forcefully and less frequently. The blood vessels will relax and dilate and thus decrease the blood pressure. Both of these acting together will decrease the blood pressure. So in this example, we have multiple effectors. We're going to see this is actually the case in several of the systems that we are going to be studying. Let's look at another example. We want to talk about childbirth. This particular image is showing a pregnant woman, and as if you could kind of see through her abdomen to see the uterus and see the baby inside. Notice that the baby is head down, and the baby's head is pushing on the cervix, which is kind of the neck of the uterus there. At full term, then you can see the baby takes up all of the space inside the mother's abdomen. It pushes all of the other organs out of the way. This is why pregnant women are so uncomfortable. As the baby's head pushes on the cervix begins to stretch. Receptors in the cervix detect the stretch and they send signals back to the pituitary gland, which is located in the brain, which we're not seeing here. The pituitary gland releases oxytocin, which is a hormone. The oxytocin produces forceful contractions of the uterus. These forceful contractions of the uterus cause the baby's head to push harder against the cervix, which produces more stretch of the cervix, which means more signals go up to the pituitary, more oxytocin is released, more forceful contractions, more pushing on the cervix, more stretch of the cervix, more signals sent to the posterior pituitary, more oxytocin released, more forceful contractions, etc., etc., etc. In this particular example, if we were to plug in these components into our scheme here, our flow of information, we would say then that the receptors are going to be the stretch receptors in the cervix. They are detecting the stretch. The control center we could identify as the posterior pituitary of the brain, which releases the oxytocin. The oxytocin is going to be detected or is going to act on the effector, which in this case is going to be the wall of the uterus, which brings about more forceful contraction. In this particular example, what we see then is that the initial stimulus, which is going to be stretch of the cervix, produces the response that we see in response to that is actually more stretch of the cervix. This is what we would call a positive feedback loop, And our positive feedback loop means that the response is actually in the same direction as the stimulus. Now here, I've identified some characteristics of negative versus positive feedback here. And the main defining factor is that it really has to do with the relationship of the response and the stimulus. In a negative feedback loop, the response negates the stimulus. In a positive feedback loop, the response is in the same direction as the stimulus. You want to be sure that you don't make value judgments. Negative feedback loops are not bad. Positive feedback loops are not good necessarily. It really has to do with the relationship of the response and the stimulus. This brings me to the end of my first little lecture. I hope you move on to the second one. Thanks very much.